Welcome back, Life Sciences. We're ready for the last segment, all right, of this session. What have we looked at so far? Remember, we're looking at the microorganism fungi, right, the kingdom fungi. And we looked at the structure, and we looked at the characteristics, and we looked at a few of the diseases that fungus, all right, could cause. A lot less severe than the diseases in our previous sections, but nonetheless might cause a bit of irritation and need to be treated. Okay, now we're going to dive into a few questions that we can ask on this section, and let's see the kind of things that the fungus can ask us. Right, guys, our first question, we always start off with a nice, easy multiple choice. Multiple choice, when I say is easy, it's not always easy, but it does. The answer is there. It gives us words that we need to start to use. So let's have a look at the first one. It says, which statement correctly describes, let's go back. I think we're going to have to get the pen going again. We always forget about the pen. Which statement correctly describes, now have a look, we've done all the microorganisms, fungi and protozoans. Now if you can remember, protozoans were the animal, all right, the animal-like structures, right, of the protist kingdom. Now let's have a look at all of our options. Number A, fungi and protozoans are all unicellular. Now that is incorrect. Protozoans, yes, are, but fungi, not all of them. So that statement is incorrect. Statement number B, fungi and protozoans all have chloroplasts. Neither fungi nor protozoans, guys, have chloroplasts. One is the animal, all right, of protists. Fungi, we know, have to live on other living organisms, so that statement would be incorrect. Fungi and protozoans are eukaryotic. Yes, they both are. Both of them are eukaryotic cells. They have a nucleus with membrane-bound structures. But we now just go and check the last question to make sure we're correct. It says, fungi and protozoans all have cell walls. Okay, fungi, yes, has a cell wall, but protozoans, being animal-like cells, they do not have at all. They just have a cell membrane. So that would be incorrect. So number C would be a correct answer. Okay, guys, the next question is one that you often are going to get in, all right, your exams or tests. And you need to read the information very carefully because it gives you options. Now it says, indicate whether each of the statements in column 1. Okay, so here they have a statement. Now you are given 4 different options, and you've got to read it carefully. All right, so your options are as follows. You are given an A and a B, as you can see over there. Now what you need to do is the following. The A and the B, all right, must, so we go to the diagram, we go to this statement, right, is A correct, is B correct? So if A is correct, you write only A. If statement B is correct, you write B only. If both A and B are correct, they both, all right, are the correct answer to the statement, you write both A and B. Or your fourth option, okay, so you've got 1, 2, 3, fourth option, neither, not A nor B is correct, then you write none. Okay, so we read column 1. It says disease causing fungus. And our 2 options are parasitic or heterotrophic. What causes disease? Parasitic causes disease. What number is it? It's A, so it's A only. There's my option, A only. Okay, guys, now the second question, all right, the description is as follows. It's a multicellular organism, right? It has branched filaments and it has a cell wall. Now, what are my 2 options? Fungi and bacteria. Now, if I look at the first question, multicellular organism, bacteria is out. So my answer must be fungi, which is A. So my answer is A only. Both of them A only. Don't get too worried if both are the same answer. Okay, now we're going to go for quite a longish question. So let's read what the question says. It says a mycorrhiza, right, which we know is a mutualistic relationship between fungal hyphae and roots. So we know it's a fungus, right, and we find it in the roots of plants. The hyphae, those are the threads, increase the absorptive surface of the plant's roots. So they make the roots bigger. Remember, we have roots and then we have all these little ones coming there and we can absorb more water. It says by aiding in the absorption of water, phosphorus, and other minerals. It's telling you important information about the experiment that you're going to do. Right, so it's telling you the mycorrhiza really helps plants to grow. The plant is photosynthetic, which means that it can make glucose and starch and provides the fungus with carbohydrates. Guys, really important, read it carefully. A lot of the information you need is going to be here. Okay, let's set up the experiment. The scientists conduct an experiment to determine, as soon as you see to determine, that is the aim of the experiment, the effect of mycorrhizal associations on plant growth. Very simply, what do they want to know? Does mycorrhiza help plants? How did I set it up? I had two groups of plants that were grown. Right? Now I'm going to change something. One group was planted in soil that had been sterilized. What does sterilization do? Sterilization gets rid of microorganisms. So in this case, right, guys, no this group, no fungus. If I sterilize it, no microorganism, no fungus. The other group was planted in the same type of soil, a fixed or controlled variable, but the soil had not been sterilized. So this group, the fungus, would have been present. All other factors remained the same, controlled variables between the two groups. The plants were allowed to grow for 8 weeks. Each week, the height in centimeters of the plant was measured. Okay, so what we have here, right, what is our independent variable? What did we choose? We choose independent variable, the soil, whether it was sterilized or not. What was the dependent variable? We must start thinking along those lines. What can I not control, right, was the height of the plants. Okay. All right, so guys, here is the results after the 8 weeks. Here we have the 8 weeks. Here, all right, we have the height of each of the plants. This was the non-fungus, fungus, and this was the fungus. So let's see what kind of questions. Number one, identify the independent variable. Now, this is quite tricky because a lot of you say, oh, it's the first column in the table. That is, all right, weeks. No, it's not. What were we looking for? What could we change? The independent variable, what did I decide? And I decided if the soil was sterilized or not. All right, okay. Independent, I. What did I choose? So if I go back to the next one, what is the dependent? Dependent is results. What was I looking for? Let's go back here. What was I looking for? I was looking for whether the plants grew or not. Okay, that was the dependent. Are they going to grow or they're not going to grow. So if we have a look at the dependent, it was the height. That's my results. What am I looking at? How well did they grow? The height of the plants. Guys, we always remember units of measurement. How did I measure it? In centimeters. Okay, always describe one way in which you ensured the results of the investigation were valid. In other words, the controlled variables. What did you do the same, right? The same to the whole experiment. Number one, what I did see here, we're going to go back to this, all right, over here. Oopsie, I'm going the wrong way. Let me go back again. Let's go back to this one over here. All right, controlled variables, look here. All right, the other group was planted the same. All factors remain the same. And here, the plants were planted in the same soil. So, what did I do the same? Same soil. Now, if I say the same factors, what do you think that means? Now I need to use a little bit of common sense. What else was the same? I'm growing plants. The same factors. Same amount of water, right? Same amount of light. And I kept them all for the same amount of time, which was 8 weeks. Oopsie, I'm going right down there. Okay, so what is it? When I find a way that I can make sure the results, that's my controlled variable. What did I do to the same groups? Okay, guys, the next one that I have to do, right, is draw a graph. Now have a look here. It says use the data to draw 2 lines. Generally, we don't use color, so if you don't use color, you need to come up with a key. Now, let's have a look how we're going to start. What's the first thing you always need to do when you have a graph, right? You are going to give me a heading. And you tell me, always tell me what you are going to draw. I'm going to draw a line graph, and what are my 2 variables? Showing the height, okay, of plants in 2 types of soil. And you can even say sterilized, non-sterilized. There I've got my heading going. Right, I'm going to draw a line graph, and I start here at 0. Now, weeks is not so much what I want to do. Yes, so weeks, that is going to be on my x-axis. All right, so guys, we're going to have weeks on my x-axis. And what am I going to have here? Height in centimeters on my y. And you're going to decide sterilized. You're going to do dot, dash, and non-sterilized, you're going to draw a straight line. Remember, I'm just going to do it randomly. You're going to point, and you're going to join your dots, and you're going to have a key. All right, so weeks, there's 1, height. Make sure that your numbers follow each other, and there you're going to put 2 line graphs on the 1 graph. Okay, guys, I'm afraid that's all we have time for. Time has run out, I'm afraid. I hope you had a good lesson. Remember, go and find that lichen. We will see you next time. Cheerio, bye.